Hello, everyone. I'm your host, Madeline Stone. Viruses are known as infamous diseases affecting our bodies, causing sickness or even death. However, recent medicine is showing that viruses can be beneficial, used to treat debilitating and deadly diseases. This week, we will be discussing virotherapy, how viruses are used to treat or cure cancer. Dr. X, thank you for joining us today.
Thank you for inviting me. I'm delighted to be here.
So, Doctor, how do viruses replicate in the first place?
So, unlike bacteria, viruses aren't living organisms and lack the ability to replicate without a host cell. In order to spread, viruses must find a host cell to do the work. The virus first binds to the surface of the host cell by attaching to the cell receptors. If the virus is enveloped, it enters the cell by fusing with the cell membrane, and if the virus is not enveloped, the virus penetrates the cell membrane. Afterwards, the virus is replicated through two main cycles, titled the lysogenic or the lytic cycle. In the lysogenic cycle, the virus injects the viral genome into the host cell. When the host cell multiplies, each daughter cell contains a copy of the viral genome. In the lytic cycle, the virus also injects the genome into the host cell, and the cell manufactures copies of the virus before the host cell ruptures, spreading more copies of the virus to other cells.
Wow, that's very interesting. My next question is, how exactly can a virus treat cancer, such as melanoma?
Well, I'm glad you asked. Virotherapy involves the usage of viral vectors. A viral vector is a carrier of genetic material into the cell. To create a viral vector, scientists take a highly infectious virus, such as adenovirus or herpes simplex virus, and remove the original viral DNA so that the patient does not get sick. Then, scientists place a specific genome, which is beneficial to the patient inside the virus. The viral vector is then administered either through IV drip or through taking tumor samples from the patient, injecting those cells with the viral vector, and returning them to the patient's infected region. Once administered, the viral vector travels to the tumor cell, latches onto the cell, and fuses with the cell membrane. The viral vector then releases the beneficial genome into the patient. The cell follows the genetic instruction to release tumor antigens, which are substances that trigger an immune response and the production of pathogen-fighting proteins called antibodies. White blood cells of the immune system recognize the tumor and release poisonous proteins, killing tumor cells. Virotherapy has already been effective at treating certain types of cancer, such as metastatic melanoma, the most serious type of skin cancer. In 2015, the FDA approved the usage of a modified herpes simplex virus called TVEC in patients suffering from metastatic melanoma. TVEC, which is encoded with the GM-CSF gene, promotes the release of tumor antigens.
That is quite an amazing medical breakthrough. I have one last question before we wrap up. What other types of cancer can potentially be treated using this method?
Currently, scientists are working to treat both non-cell small lung cancer and small cell lung cancer using virotherapy. Recent studies involving a lysogenic adenovector known as ADP53 have suggested that this viral vector is able to treat those cancers. A study conducted on 58 individuals showed that patients had few negative side effects from the treatment. However, more studies will be needed as the effectiveness of the treatment varies from patient to patient. Scientists hope that AD-P53 will reveal more promising results with the ADP53 treatment for non-cell small lung cancer in the future.
Now that is an excellent news story. I want to thank you for tuning in to AP Bio News and thank Dr. X for speaking to us about this. This was AP Bio News. Stay tuned for more and stay curious.